So good afternoon, everyone. Welcome to the April edition of the NITheCS of NITheCS. Yeah? So this month, Govender with us as a speaker. Krishna is a senior researcher at the CHPC, the Center for High Performance Computing, and he's an expert in computational chemistry and material science. And at the CHPC, he supports on the CHPC clusters. Yeah. Krishna has also an associate position with the Department of Chemical Sciences at the University of Johannesburg, where he also supervises masters and collaborations across the country and international engagement with the CHPC, summer schools, winter school courses and training in Python, Linux, and so on and so forth. Yeah. So Krishna, thank you so much for joining us and for offering this very interesting course in computer-aided drug design. You know, we are all, since COVID, we are all very sensitive to issues related to drug design. So it will be very interesting to learn all of in Zoom technical language. So each of you has the right to unmute and ask questions at any time. Yeah. So please do that in a reasonable way so that we don't interrupt the lecture too much. But the idea is to keep it interactive so that Krishna can assist us in learning something about how to use Schrödinger for drug design. Yeah. So Krishna, please, you're most welcome.
Okay. Prof, can you still hear me? Prof, can you hear me?
Yes, I can hear you, but we still can't see your screen.
I'm going to start sharing now. Just give me a
Okay, perfect, excellent. No, sorry, I was just worried, you know, sometimes the internet can freeze or something. Excellent.
No, always.
Yeah, now it's there and we can see it. So you're welcome to start, Krishna. Thank you very much.
Okay, thank you, Prof, for the introduction. So, guys, as Prof has mentioned, we want to try and make this very interactive, right? So I'm going to actually now put the chat box on another screen of mine here, together with the participant list. So if there's at any point that you want to ask me something, you can put it in the chat or raise your hand and I'm happy to stop and then we can just answer your question. I don't want to just be lecturing the whole time and then nobody's really following what's happening. So I would much rather have it to be an interactive thing. So please stop me at any point, right? So Prof has already told you who I am. I'm a research scientist at the Center for High Performance Computing. And what I do is I usually do research in computational chemistry and material science. And from the computational chemistry perspective, I do simulations dealing with drug design, right? So this is one of the things that I'm going to try and introduce to you guys today. Just those of you that might already know most of this stuff, it is a beginner course. So just a heads up, I am just going to be introducing some very basic concepts. So those that have already done some simulation work, this might not be that interesting for you, but maybe I can teach you one or two things. Okay, so let me just get started. Right, so this is going to be the overview of my talk today. We're not going to be doing any hands-on stuff today. I'll only start with the hands-on interactive stuff from next week on. So we're just going to try and go through this whole process of how do you go about designing a drug, what's the procedure you need to follow, and so on, right? So this is the overview that we'll be using. So why would we want to develop drugs? And why do we want to do drug design? So the old way of doing it was to produce drug candidates that was primarily done by a medicinal chemist in a lab, right? So often it used to be a trial and error thing. That look, you try something, it works, you try something, it doesn't work. And as it says there, I've just found 10,000 ways that won't work, right? So you can find multiple ways where it won't work, and very often at some point you will get the right answer. So this is then where some of the compounds were found by either mistake or they're found by logical guessing. Maybe it's some sort of normal process that people follow. They use some data or some information, and then they try to guess what ligand would work best or what drug would work best. Now, these modifications are often done by guessing, as I say. And yes, so one can imagine if you're doing this, it's going to be a time-consuming process, right? And on top of that, it's a lot of chemicals that one needs to make use of. So it's going to be very costly as well. So what is the modern way that one would like to design drugs? If you consider the modern way, similar to how you would look at something like a GPS, right? You know where you want to go, and you just need to be steered in the right direction. And you use the computer to provide you with that information that will allow you to analyze the data and build some hypothesis that can help you to get the desired result. So this results in you finding a solution faster. And due to the low or no cost, you're basically saving a lot of money, right? So this is the drug design process. Let me just quickly get my pointer. Right, so we start off with, so typically it's like a 3-phase process where you have your hit compound identification, the clinical trials, and your preclinical trials. So preclinical, then clinical trials, right? So your target discovery is where you want to find some sort of target of a molecule that's integral to the gene regulation or intercellular signaling, right? And you need to ensure that this target is druggable. So whatever you identify in, say, a protein or whatever it may be, it needs to be able to take a drug, or it must accommodate a drug or a drug candidate, right? So from there, you get to the target validation. So say we found something that's druggable, now we want to validate it. So you validate the hypothesis by testing the antibody interactions, and you modulate the drug activity to target changing molecular structure, right? You start changing the structure up, see if it does something different, or does it do the same thing? Is it interacting with what you want it to interact with? From there, you then go to the lead compound identification, which then tests the drug mechanism of action. You identify the safety tests and conduct a cell culture testing. You also look at the pharmacokinetics and pharmacodynamics. Now, what happens at this point is that sometimes you design a drug, and of course it works, but then the problem is it might have side effects, right? So you want to try and avoid those side effects. So you have to often modify that drug to make sure that it doesn't have adverse effects on things that you don't want it to be having effects on. So once you've got that, you can do the optimization of your lead compounds, which you then alter the design of the molecule to prevent, as we said now, off-target effects. You optimize the dosage, and you also determine now, how would you want to give a patient this? Do you want to give them orally, or is it going to be via some injection, right? Then you conduct tests for the drugs updated by 3D cell culture systems. We then do the preclinical development. If all of these phases pass, right, so you get to the preclinical development. Now in this preclinical, you have to test the drug in the alternative cell lines and in vivo. Most commonly, people use mice or rats in research. So when they're trying to test these things, and then you plan for either small or large scale production if approved, right? Then the final step is the FDA approval. That's where your clinical trials comes in. That's the longest process from all of us. And as you see there, it's like 6 to 7 years, right? So your new drug is approved. You then make enough of it. You then start getting into the human phase trials. The FDA conducts reviews to check that everything is working fine, and it approves you after the phase 3 clinical trials. And then you continue monitoring for dosage and safety. And that's it, right? But as you can see, this is not a very simple process. It's not just like you develop a drug and it's in the market and you're done. You have to go through a stringent process before you can actually get this drug on the market. Okay. Okay. So what exactly is a drug? So if you consider small molecules, right? So in chemical terms, a drug is typically a very small molecule that would bind to a target site. So in this case, in the cases of biology, we usually look at things like large proteins or enzymes, which are considered your locks. And then this small molecule that goes into this protein or enzyme can be considered the key to the lock, right? And it's used to turn on and off certain biochemical processes in the body. So before we continue with this whole lock and key story, I would like to quickly show you what is a protein, right? So what exactly is this protein that I'm now talking about and that has this target site? So protein is nothing more than just a building block, right? It's a block of many different amino acids and which are small. So and amino acids are typically small organic molecules. And there's a carbon in the center. I'm going to show you just now what the molecules actually look like. So let me just quickly go to the next slide. That will help. Right, so here we go. We've got a set of amino acids. There's 20 of them in total. And these amino acids can actually bind to each other in various different sequences. Right, so you can have alanine with leucine and leucine with isoleucine and then back to valine. And you can have a long chain of all of these things put together, right? So if that happens, you end up with something like this. You've got a primary structure where you've got a bunch of amino acids all bound together. And each of these amino acids has a side chain, right? Which is this R group. So this is one amino acid, there's another. In this case, it's only 4. But of course, the chain can be as long as the protein wants to be. So if you have this, you then get a long straight chain, which makes up this protein structure. But this chain doesn't stay straight. What happens is this protein starts to fold and you get what's called a secondary structure because these R groups start to interact with each other. So this R group might want to interact with that one, this one with that one, and so on and so forth until you eventually get this nice swirly curved thing, which is essentially a protein. So when a protein forms inside your body, typically they don't remain straight. And often you'll hear things like people talk about protein folding or something along those lines. And depending on how certain proteins fold, the interactions that you get in the body actually can change, right? Depends on which side the protein folds. If it folds to the left, folds to the right, it can give you different types of interactions. Okay, so sticking with the what is a protein, what exactly can proteins do? So proteins in your body actually can do a whole bunch of stuff. So the proteins typically have antibodies, right? So the antibodies are like the ninjas of your body, if you consider that. So basically they fight off disease, right? They try to fight off infection or any disease that you might have. And if your antibodies are strong enough, you can fight off lots of diseases. Unfortunately, something like COVID, a lot of people's antibodies weren't strong enough to fight it off, right? That's why we ended up having to get a vaccine for it. But then there's an enzyme. Enzymes are typically used to catalyze chemical reactions. So when they catalyze the stuff, it typically means that, so if you, a person that's doing chemistry, you would know that catalysis is nothing more than speeding up a reaction. So an enzyme, that's exactly what it does. It speeds up certain reactions that happen in the body. And if it wasn't for the enzyme, maybe a reaction would take like 300 years to happen and you'd probably be dead versus taking like a few seconds to a minute. So this is the type of things that enzymes do, right? So they're quite powerful things. Then you've got messenger proteins, which transport messages. So in this case, you can take types of hormones and you transmit the signals to coordinate biology processes between the cell, tissues, and organs. Then you have a structural component. The structural component provides structure and support for the cells, right? On a large scale, they also allow the body to move. So structural is typically those things that make your muscles move, and the muscular component, that would be the structural components. And then transport. So just like with Uber, our body also has its own Uber. So it transports certain things from one part of the body to the other so that it can go do the job somewhere else. So this is typically what the transport proteins would do. They carry cells or drugs even from one part to the next. Right. So let's get back to our locking key now, right? So if we look at our locking key, how exactly do we, from this large box of keys here, because that's exactly what our drug candidates are, it's just a big box of keys. How do we find the key that will fit into the lock? So your key here in this case is the substrate, right? That's your drug. And then the lock here is the protein. So typically you want the key to fit into your lock perfectly, right? So this protein has this lock and there's the key, so the key fits perfect. You also have cases where there's incorrect substrates. In this case, what might also happen is it might never actually go into the active site. So this dotted region here is our active site. Here the picture shows you that the key is inside, but in most cases, when the ligand is not good enough or the drugs are good enough, it won't even go into the active site. So it's just a complete dud. You can throw it away. So we need to try and figure out how can we actually find the key from this maze of keys. Right, so before we get to our keys, let's quickly check. How does one actually do this sort of identification of drugs? So experimentally, they nowadays use something called high throughput screening. So as you can see here, this is the type of machinery. So it's an automated process where you have a bunch of these petri dishes and you've got samples that sit inside each one of these that get injected into these dishes. So each one of the dishes might have your protein, and then the samples have different ligands that are there or different drugs, and then you test them by putting them into the sample trays. But of course, this is using robotics, right? So, I mean, you can imagine if you are experimentalist and you have to stand there with a pipette and pipette into each one of these things, you're going to sit there forever. And at one point, this is what was done. But obviously now with technology, things have changed. Right? So here they use a robotic process and they can sample hundreds of thousands of different ligands and see how they perform. Right? So the biggest thing here is that you can imagine if you've got all of these ligands and all of these chemicals that you need to put into each one of those sample things before you inject it, it's going to be costly, right? There's definitely a money aspect involved here. So what can we use as an alternative? We can use something called virtual screening. So instead of high throughput screening, we use the virtual screening process. Now here, we're doing very similar thing like what they do in the lab, that we're trying to inject these sample ligands into our different proteins, but without actually having the physical chemicals for it. All we need is the chemical structures for the drugs, and then we can take all of those drugs and dock them and try and see what happens, right? So this is just a computational technique that allows you to do this process, and it allows you to do it on a much larger sample size. So as opposed to hundreds of thousands, you could probably do millions of drug candidates at the same time. Okay, so if we have a look here, so this is, yeah, here we go. So say, for example, we were in the lab, right? And we wish to actually now look for a certain drug. But the problem is now that when we look for a certain hits, maybe we have drug candidates that only go up to this point, right? And when we do it experimentally, that's all the ligands we sample. So these are all our hits. However, what if the drug we were interested in is actually here in this yellow? You're never going to get there, right? Because if you sample in ligands that's only here, we're never going to get to this point. So what do we need to do? We actually need to, this is where the power of computing comes in, is that we need to sample a lot of ligand phase space. And this is the biggest challenge. Experimentally, you cannot do that because there's just too many samples. It's going to cost you too much money. But from a computational perspective, it is possible for you to sample, say, a larger region of the space and dock all of them and eventually land here saying that, look, this is actually the right region where the drug is and the potential drug candidate. So often when it comes to doing this stuff computationally, people, most of the time they get lost, because I know most supervisors tell you, look, you need to go and do docking, go and do it. So you don't know where to actually find these ligands. So this is why I provided you with a table here, and I will be sharing the slides with Prof as well at the end of my presentation so that you guys can have it. And then there's links to all of the sites that actually provide different ligand libraries. And as you can see, there's large libraries. I think when I last checked, if I were to ask you to guess, right? So let me ask you something here in the chat. Give me a number. If I were to ask you, tell me how many organic compounds can be made from pure organic ligands. Just give me a guess, a wild guess. Come, there has to be somebody. There's 64 people in this, so there must be something. Okay, 110. Okay, 200. All right. Okay, so the numbers are going up. I see. There are too many combinations. All right, Franklin. Okay. Okay, so when last I checked, the total number that's possible is 10 to the power 63 different organic compounds that can be produced from just pure organic molecules, right? So it's obviously larger now. I think I checked this like a year or so ago. But you can imagine that's a large number of compounds, right? So just like if we look at this figure here, just this small piece here could be like a couple 100,000 organic ligands and a few 100,000 here. And then, I mean, this whole thing here is like, say, 10 to the 63. So it's ridiculous. There's no way that you're possibly going to sample all of this in a laboratory experiment. You probably can't even sample it in a computational experiment because there's just not enough computing power to actually do all 10 to the 63. We're getting there, but maybe not just now. Okay, so what exactly is computer-aided drug design? So I've been now harping about how people did it experimentally, and now how can we do it differently? So there's two general types of computer-aided drug design, right? And yes, I know it's called CAD, which is very similar to the engineering application, which is also called CAD, but this just has an extra D. So this is the two approaches that one can use when doing the drug design is you have a structure-based drug design versus a ligand-based drug design. So what exactly is the difference? So for the structure-based drug design, you need a 3D structural target, right? So you need a protein structure and you need to know this protein structure. It needs to be crystallized out. So you need to have enough information on the structure so that you can use it to then start looking for active sites, for example. So in this case, if the 3D structure is known, then we can do a structure-based drug design. However, the 3D structure sometimes is not always known. But before I get there, with structure-based drug design, if you know the structure, you can identify the active site. You can then do something called molecular docking, right? You take the various different drug types and then you put them into the active sites and you see what the interaction is once you actually dock them in the active site. Then you have the unknown, right? So if we don't know the 3D structure, then there is a technique which is known as homology modeling. This will allow you to build up the amino acid sequence so that you actually build a 3D structure. So if you can build this 3D structure, you end up back with the structure-based drug design because you started off with maybe some sequences that were missing, and then the software is able to rebuild the sequences and eventually get you to a structure that looks good, which you can then use for molecular docking. Right, then thing is, that's not always the case, right? There are structures which you don't know at all. So if you cannot use homology modeling to build the 3D structure, you're going to have to go with what's called ligand-based drug design. So the ligand-based drug design, there are two different approaches. There's one where you know all of the active ligands and what their activity is. So if that's the case, then you can use things called singularity searches. pharmacophore mapping, or quantum structure activity relationship, right? So the singularity searches are usually used for a small number of compounds, like maybe 1 or 2 ligands. The pharmacophore mapping is when you have several ligands that are similar. And then the quantitative structure analysis, then that you're using certain quantities to identify what's the best ligand that you can use. And then what you do is, from designing a ligand, you can then take that ligand and dock it against various different targets, as opposed to just docking it onto one target to see how effective it is. And then if you have your active and inactive, that you know certain compounds are active, certain compounds are inactive, then you can switch over to the machine learning approach of doing things, right? So you can then let the machine take all of this large library of ligands and sort them out based on the activity and inactivity, and then show you which ones would work best. So let's now move on to the docking parts, right? So what exactly is docking? So docking is nothing more than just predicting the optimal geometry, or the orientation and conformation of interacting molecules in space. And it shows you the stability of a complex that's formed when you actually put a drug into the active site. So these are the different types of docking procedures one can get, right? If you have an antibody, you can dock an antigen onto the antibody. If you have an enzyme, you can dock a substrate. If you have a transport protein, you can have certain molecule that's going to be transported on this transport protein. And then if you have a receptor, you'll put a signal processing drug onto that receptor. So there's different approaches when it comes to docking, right? We've seen the locking key. I've showed you guys that, look, there's a bunch of keys that we can actually choose from. And then the lock is then something we need to try and figure out, right? We have to figure out whether the key fits in the lock. So this is the typical lock and key approach, where you have your drug, your protein, and then the key fits into the lock. Simple. But not all of the procedures are that simple. You also get something called induced fit docking. So here, both the protein and the ligand can actually, say, morph, right? So they're able to change shape and change conformation and try to fit each other. So essentially, it's like this key is sort of adjusting itself so that it fits into the lock that was present. So you see the shape of both the protein and the ligand, they both change. And then you get conformational ensembles. And this is one of the more computationally intensive techniques. So basically here, what you're doing is you look at various different conformations of your protein, and you dock the ligands into each one of these different conformations. And then you find some that where the ligand interacts well, you find others where the protein is actually active in that particular conformation, some where you get a substrate that binds, others where it's just inactive, there's no activity at all. But this is all from the same protein. It's all just a matter of the different conformations that it forms. Right, so when you do the docking, you get you've got your protein, you've got your ligand, and the ligand goes into the pocket, which is called your binding pocket or your active site, and it interacts with its surrounding environment, right? So here I have an example of what happens during the docking process. So when you usually get a structure, in some cases, the crystal structure will come with ligand bound to it, right? This doesn't always happen, but in some cases, it crystallizes with the ligand bound, so you can see where the active site actually is. So these are the sort of easier proteins to deal with, because you already know where the active site is. So this case, this purplish one here is basically the ligand that came with the protein. And then the one that's busy moving around there is the docked ligand. So what the software does is it tries to dock this ligand into the best conformation that it can predict. And as you can see, it's not too far off from the experiment, right? So you see, it doesn't lie too far from the experiment, which means the docking is actually going quite well. So if you do docking, typically this is something that you would want to do. If you have a crystal structure with the ligand, you redock the ligand and try and see what is the RMSD distance. So what is the root mean square deviation between the ligand that's docked versus the ligand that's already there? And you want to make that as small as possible to know that your method actually works well. Right, so the different docking categories that we have would be in protein ligand. You also can get protein-protein docking. There's protein nucleic acid, enzyme substrate, and then ligand nucleic acid. So your protein-protein is quite complicated because you have two massive proteins and they actually then need to change conformation and dock into each other. And the protein nucleic acid also. The better understood ones are the protein ligand docking studies. Right, so if you look at this graph here, this just shows you the level of complexity. So we go from both being rigid, which is the easiest to do, where none of the conformations are changing at all. Then you've got your ligand that actually can change conformation before it docks into your rigid protein. Or you've got the protein that can be flexible, the ligand's rigid, and then the final one is where both are flexible. So that's the most complicated one and the most computationally intensive from all of it. Okay, so of course, this is a NITheCS lecture, so there has to be a little bit of physics involved somewhere. So remember, this stuff is all built on the concept of physics, right? So typically, the methods that we use in sort of drug design go one of two routes. You're either going to do it via an empirical route, or you're going to do it using quantum mechanics. So the empirical route, you don't care about the electrons. Right? You're neglecting the electrons completely. You're like, no, you know what? Electrons, I don't know. What are those things? So what happens here is you actually have parameters for each and everything that you're doing here. So all your atoms are represented by balls, and anything between them is represented by a spring, right? So for bonds, this spring here basically is just giving you the length between these two atoms, so the bond length. Then for angles, you have the bond angle. That's a parameter. You also get improper dihedrals, torsions, which is your normal dihedral angles, electrostatics, and van der Waals. And your van der Waals are typically done by something called a Lennard-Jones potential. Then you get the other end, which is the quantum mechanics. And one thing I should mention here is, remember, these are all parameters. So the biggest problem you have with doing this is that when it comes to doing a reaction, you are not going to be able to do it, because what you cannot do is make and break a bond when it comes to molecular mechanics. You have to do that quantum mechanics because that's a quantum sort of property. So you cannot do breaking and making here, right? You can just see how those things fit together. That's about it. Then you've got on the other end, which is obviously a quantum side. So of course, as all of you should already know, this is solving the Schrödinger equation, right? And there's different legs of quantum mechanics. There's your semi-empirical, which is quantum, but there are parameters involved as well. So you actually do a parameterization, similar to how you do it for force fields, but it's quantum mechanics. So you're still solving the wave function, but you're neglecting some of the electron correlations so that you can actually get these different methods. And the problem with this is that it's usually very specific to the class of ligands or class of molecules you parameterize for. So if you don't have a certain thing in your parameter set and you want to model something outside that parameter set, this is probably not going to do the job. It's going to give you better results. So that's why you need to go to things like ab initio and DFT, right? Because here you're counting for all of the electrons. And in that case, you can then model anything. However, remember, computational costs goes up substantially when you move from left to right. So from molecular mechanics to, say, ab initio and DFT, there's a dramatic increase in the computational time. So one has to just keep that in mind when doing this stuff. Right, so let's get back to our docking. So how exactly does this docking work, right? So if we start with the protein structure, so this is a crystal structure that I've got from the PDB data bank. That's another one that you should keep in mind, because whenever you're looking for any proteins, that's the first place you should go, is the PDB data bank. So that gives you 3D structures of your proteins. So this is a protein structure which is downloaded. Then after you get the structure, you have to obviously prepare the structure. So once it's prepared, you'll notice that this structure I have actually has a ligand bound to the active site already. So I already know where the active site is, right? So what I can do is when I prepare it, I then remove the ligand and I generate what's called a grid around that region, which is considered active. This green region is the region which is the active site, but the purpley region on the outside is just gives your ligand freedom to move up to a maximum of that section. But if the docking protocol works well, it shouldn't go further than the screen part, right? And you'll end up with the ligand binding here somewhere when you dock. Right, so now, once you have your grid, you need the ligand library, right? So you say you have a ligand which you start with fine. The first approach would be to take that ligand which your crystal came with, redock it, and see if it works well. When you're happy with that, you then go to one of those sites that I showed you earlier, and you go and look for ligands that are either similar or have similar structural activity or anything along those lines. And you get like a bunch of ligands that are within a certain class. And you then take those ligands and you prepare so that they're actually ready to be used for docking. So once the preparation is done, you take the grid and the ligands and you dock the two together, right? So then the ligands then start changing conformation and then they dock into a specific position where they're most happy. And from there, you end up getting something called a scoring process. So this scoring is done more on an enrichment process. So the way Schrödinger does it anyways is that based on how the thing fits inside the active site and how it interacts with its surrounding environment, things like hydrogen bonds, van der Waals interactions, these all contribute towards the score to make it more favorable in a certain position. Right, and then finally you do your post-processing, where you take the squad ligands and you go through them and see, is it looking like something that's reasonable, or maybe you just went the wrong direction? Because sometimes happens, right, that maybe your ligand library is not the most appropriate for what you're trying to find. So you then have to go back to the drawing board and try and redesign your library, or maybe add more compounds to your library. Right, so post-docking. Now, when you're done with the docking, you take the hits, you can synthesize them and test them experimentally. That is one thing you can do. However, that's not always the best approach to use, because the thing is, even if a ligand is docked into an active site and it actually interacts with an active site nicely, there's a problem that it might not be the best drug to use for that active site. So what you should be doing is running further simulations to verify that that possible hit is in fact stable inside this active site. And what does this mean? This essentially means that you need to try and mimic the environment in the body, right? So the best way to do that is to solvate your system, because as you know, water has there's a lot of water in your body. So you need to solvate the system, and then you have to run molecular dynamics, where you're taking actual time into consideration and the movement of all of these atoms in the protein and in your ligand, and you're trying to see if the protein moves for a certain period of time, does the ligand stay bound to the active site, or does it actually get kicked out? Now, of course, ligands getting kicked out is not a bad thing. It is something that sometimes you do want, especially if it's in a transport medium. So you have to look at the results and base it on what you actually trying to find. If you're finding a ligand that you want to be bound on the active site, it needs to stay there. It can't just, during the simulation, just pop up, because that means it's not a really good candidate. Right, so it's often used to explore conformational space, right, to molecular dynamics. And in this approach, it's if you look at the conventional docking approach, that's a single point model, right? Your protein might be a fixed structure. So the protein is fixed and the ligand allows slightly, and the ligand binds to this thing, but the protein has been fixed the entire time. So you could consider that more a single point model. Versus here you're allowing everything to move. So the whole nuclear system is forced into motion. So are you taking things like motion into consideration? And it assists in determining if the ligand stays bound to the active site, as I mentioned. Right, so as I say, getting a ligand popping out of the active site is not necessarily a bad thing, right? So in this particular case, this is actually a transport protein. And the simulation here Let's hope it plays now. Yeah, there we go. So a water box around this thing shown. So during the course of the simulation, this drug moves around in this protein. Then the protein gets to its destination, and when it does, it actually folds and kicks that drug out. So it kicks that ligand out. And basically, what's going to happen there is that ligand then going to go to its destination, right, which is the next protein structure where it can do the job. Now, what would be cool is if one could actually simulate that whole process. So here's a challenge for you guys. If you want something to do research-wise, you often get these heat shock proteins that are considered transport medium. So you can have a heat shock protein, and then in the cell, you have another protein next door to it. But it would be nice to see if via dynamics, you can actually see this ligand move from the one protein to the other and bind into the active site. It's probably going to take quite a long time simulation-wise, but it would be some great stuff to actually have if you can do it. Now, what if you have a reaction in your active site? So I've told you guys about the enzymes, right? And say there's a reaction taking place in the enzyme, which is what happens in enzymes. So here's an example. We, of course, won't be covering this in this course, but I just wanted to show you guys what the potential of this computing is so that you can have an idea for yourselves. So this is PNP, right? The purine nucleoside phosphorylase, right? So it's got these three things here that make up this triangle. And in there is the ligands that are sitting in the active site. And this is an enzyme, right? So this speeds up this process that's going to happen inside here. So this is the reaction that's proposed for this particular system. You've got a phosphate, and then you've got this nitro group. This is going to be the breaking carbon. Let me put my laser pointer on again. So this will be the breaking carbon, and this is the bonding carbon, right? So there's one bond being broken, one bond being made, right? And then this is the product that you expect. So one way one can do this is you can simulate the thing up to a point using molecular dynamics, but then obviously the reaction's not going to happen, right? It's going to, the phosphorus is going to stay in its own space, the nitrogen is going to stay in its own space because the bonds cannot be made or broken. So what you can do is you take that whole protein structure with those reactants that are sitting there, and you run it through something called Onium. Onium is a method which is developed by Gaussian. You could probably do that. The one issue I have with that is that you, again, forcing your protein to be static. So your protein is going to stand still the whole time while this reaction takes place. So yes, it will give you something like your barrier height, and you'll be able to determine that. But the problem is you're not taking the interactions of the protein into consideration. So because it's not taking that mechanism into consideration, a better approach to use is something called QMMM, MD, right? So this is actually something which Martin Karplus, Arya Warshel, and Michael Levitt won the Nobel Prize for in 2013, where they're actually able to take the QMMM and MD and do simulations with that whole process, where you have a certain region you will treat quantum mechanically, the rest of your system is treated molecular mechanically, and then you also bring in the motion of the molecule into consideration. So let's see, how would this work? So the PNP's active site, these are the amino acids that sit around the active site, right? So we want to take most, if not all, of these amino acids into consideration. The thing is, when I ran this, it was a couple of years back, and obviously the technology wasn't there for me to run it for a very long time. So I had to make do with cutting down some of these amino acid residues. So I did cut it down, and eventually, okay, fine. So this is the mechanism again, right? But what happens here is we want to do this reaction inside a water medium. So you want to have water and all of the interactions taken into consideration. Now, what I want to ask you guys is, if you were to do a reaction like this, which bond is being broken and which bond is being made? So you can give me in the chat. I'm watching my chat. Nobody's been asking questions the whole time. I hope you're not sleeping. So what bonds being broken and what bonds being made? If you look at this. I'm going to give you a minute. Okay, carbon-nitrogen bond breaking, fine. So 1,9 is the breaking bond, okay? And then the bond being made. Carbon-hydrogen. Okay, not carbon-hydrogen. Carbon 1 forms a bond with O from PO4. Yes, so Darren, that's correct. So you've got a carbon-oxygen bond forming. You've got a carbon-nitrogen bond breaking, right? So of course, what would we think are the reaction coordinates for this? The same things, right? The same thing where the bond is being broken and the bond is being made. What if I were to tell you that this one, even though it's a carbon-oxygen being formed, that's not the reaction coordinate? What would you say? Would you believe me? You'll say I'm lying. Yes, I see somebody wants to say something. Come guys, so if I tell you that this carbon nitrogen is the bond being broken, but the second reaction coordinate is not this oxygen carbon. What do you all think it would be? N9 forms double bond 2. Okay, when it breaks off, yes, it would, because remember the delocalization, it would. So we see it does form a double bond. What we also find is that the other nitrogen actually grabs a hydrogen from somewhere. But what I want to see is the phosphate. So this phosphate, what do you think? If not that oxygen, what would be the reaction coordinate? Anybody? I'm going to give you a minute. Maybe a water molecule involved somewhere. Okay, good, Ephraim, fine. Maybe there's a water that comes in from somewhere, because remember, this environment is surrounded by water, right? So there could be water, because remember, there's also, when it comes to proteins and enzymes, you can have those that are actually water soluble. So they're hydrophilic. And then you also get those that are hydrophobic, that push water away. So maybe water, fine. Anyone else? So if I exclude myself and Prof. Francisco, that's 67 people. There must be somebody that can give me something. Okay, let me put you out of your misery, right? Look at this. There's an O here, it's got a negative charge, right? There's an O here, it's also got a negative charge. And then there's a double bond O. Now, those of you that do chemistry should know that negative charge can be delocalized here, and this double bond also can be delocalized. So if there's a delocalization happening, you've got 3 oxygens that have the potential to bond to that active set. How do you know which oxygen is the right oxygen? So when it comes to making that bond, how can you tell me that it's definitely that O and not this O that's actually forming the bond? Dipole moment, fine, but now if you consider the fact that if you're treating this thing quantum already, right, the dipole should be taken into consideration. Nobody? Electron density, okay, that's also an option, fine. But as I told you, if I don't know which oxygen is going to be bonding to that carbon, then does it make sense for me to use O as a reaction coordinate? Because aren't I being biased if I choose a specific oxygen? So let's see. That's my reaction coordinates. The nitrogen is being broken. Instead of using any one of those O's, I use phosphor as my reaction coordinate. Because remember, when the phosphor gets close enough, what's going to happen is the oxygen that's the most appropriate to that carbon is going to be the one that's going to bond. So you shouldn't get a bond with just any oxygen. You'll be able to sample more phase space and you'll be able to determine which O is the best O to bond to this to that carbon atom. And here's where we made use of QMMM, right? So you have this part is QM region, the MM was the rest of it. But because these amino acid residues had to be chopped, you have to use special methods for that. So there's something called the GHO method where you actually put an atom in between to represent the bond between the QM and the MM region. And so when we do this, what if you sample which O has a lower exactly? So if that's 100% correct, you could sample each oxygen and find out which one has the lowest barrier. Or you could sample just via the phosphor and see which one has the lowest barrier. I'm going to show you now what the dynamics looks like. So if you consider those two reaction coordinates, right? There's our reaction coordinate with the nitrogen. When that bond stretches, this bond gets smaller. So here, this black region is your transition state. So this is where you cross over from here to there. The transition state sitting about here. And when we look at that transition state, just the region of the dynamics where that transition state sits, we find, well, I actually sampled for a little bit further, just so that I could see what was happening. When I look at the different oxygens, so there we've got O4, O2, and O3. So O4 is the one that samples the most because it's the one that actually forms a carbon-oxygen bond the most. So O4 is actually the oxygen that seems the most relevant. The other two also do form bonds, but they don't form bonds for as long as oxygen 4 does. So in this case, oxygen 4 is then the ligand that you want to use. But the nice thing here is that because you've sampled all of these, when it comes to that specific reaction, you know exactly which oxygen, and you can then say, okay, look, O4 is the one that wants to bond to this carbon. And so then let's see, what does it look like? With O4 in the transition state, there's the region between the carbon and the oxygen, and there's the carbon-nitrogen bond. You'll also notice that this nitrogen actually picked up a hydrogen from somewhere. It would have probably picked it up from maybe a water that's sitting here. And our reaction said that there should be an H bond to this nitrogen, right? So we know that that's right. And so yes, so this is what the transition state looked like when we did this. So just to sort of round up here, what sort of software can you make use of to do these different types of studies? So Schrödinger is one. And for the purpose of this course, we will be using Schrödinger. But there's also Amber, there's GROMACS, there's NWChem, Gaussian, OCCA, DMOL, NAMD, Autodoc, Charm, and then your visualizers are VMD, GaussView, and Avogadro. GaussView is licensed, but VMD and Avogadro are free. So those are things that you can visualize your molecules. Now, as a final thing, why do we need HPC for this stuff? You can imagine if I have a ligand library that's millions of compounds, how long is it going to take me to dock all of those millions into the active site of a protein on my laptop? It's going to take very long, right? You're going to be sitting there for a long, long time. And you don't want that. You want it to process quickly because the whole point of doing it, the computational route, is to be faster than experiment so that you can get results out and tell the experimentalists, look, this is the route that you need to go. So this is where you need the power of HPC because they give you access to much more resources and you can run over many more cores and you can then sample this ligand space a lot faster. And once you have possible targets, you know you want to run molecular dynamics. Again, running that on your laptop is going to take forever. So because of the latest technology we have, you want to rather use GPUs, and you have access to that in the HPC environment. So access to the software. I'm going to quickly just walk you guys through this. Let me stop sharing the screen. And I'm going to just go to that site. Sorry guys, 1 minute. I know my time is like almost up.
No, there is no rush, Krishna.
Okay, I'm just going to quickly show you guys how you can get access to the software so that next week, when we do start hands-on stuff, you guys actually have the software. So you should be able to go to the site, which is users.chpc.ac.za. I'm going to log out of this just to show you all how you actually get access, right? Right. So you'll end up on this page once you come here. Those of you that are permanent academic staff members at your institution, whether it be our SKA partners or South African academics, you all can apply to become a principal investigator. And then the students, you guys will apply to be users, right? Once everything is done, then I need to make mention here that unfortunately the Schrödinger license is only for South African academics. So when you do send me a request, I'm going to check to make sure that you are part of a South African institution before I can give you access to Schrödinger software. So for those of you that don't have access to the license, there are open source packages. Unfortunately, I'm not going to be covering it in this mini-school, but maybe something I can look into for the future. But all right, once you actually get an account, or your supervisor will get an account, they'll be able to create what's called a research program. They can then click on their research program, and then they can add you as students to their program. So I'm going to quickly show you how that's done. And then once that's finished, your account will be created on the cluster. And when that happens, you then send me an email to say that, look, you want access to Schrödinger. And then from there, I'll take it from there and give you the access that you need. But let me just wait for this to load quickly so I can show you how to add people. Oh, and supervisors, if you want to be a user yourself, you need to also add yourself. Because if you don't add yourself, you don't have an account on the cluster. So once you click on your program, you go to edit program, and then you choose from a list of people who you want to add to your program. So you repost access link here. Okay, I will. There, that's the link. Okay, so you get to your research program. This is what it looks like. And then from a drop-down menu, you'll be able to select your students, and you can select yourself also. And then you add you. When you're done, you go to the bottom and you just save it. And then basically, once that's done, our administration on the CHPC side will then get an email to say that these accounts need to be created, and then they will create an account for you. And once your account's made, then you get in touch with me, and I will then be able to give you access to the software. As a last thing, let me quickly just go back to my presentation here. Right. Okay. So just as a final thing, I would like to thank Prof for actually giving me the opportunity to do this with you guys. And then the CHPC and UJ, where I actually do most of my research. And then thank you guys for listening. So if there's any questions, now's the time.
Yeah, thank you very much, Krishna, for this great introduction to computer-aided drug design. It was really very, very, very instructive, and I guess we all learned a lot. And also, thank you very much for reminding us about the procedure to set up a login on the CHPC, so that from next week we can we can start playing with Schrödinger and discover our own drugs. Yeah. So no, thank you very much, Krishna. I think we had already a few interactions and questions and comments during your presentation, but please, now is the time for the participants to ask more questions if they so wish. Please, you are all able to unmute yourself and ask the question. Just try to follow some kind of procedure so that we don't speak all at the same time. Yeah, please, if you have questions, just ask.
Always just raise a hand if you want to ask a question.
Yeah, that's also possible. Yeah. I think everybody is scared. So there is a question in the chat, and that is about if people, once they have registered with the CHPC, do they have to download Schrödinger or do they have to use it on the CHPC?
So when it comes to the actual interface, which is Maestro, that's what you will be using. I will send you the download link. Once you actually register with CHPC, I'll give you the download link. And if you've got a Mac or a Linux, then I'll direct you to where you get the download from.
Excellent.
And next, I'm going to take you through the installation process, because I know a lot of people have problems when it comes to installing it, because there's a hosts file you need to edit. So I'll take you step by step through that whole process.
Excellent. That will be very useful, and I'm sure that then next week we will have many more questions. Yeah. No, fantastic. Sorry. Here comes a question by Benjamin. Benjamin asks, what type of COVID drugs were or could be discovered through the CHPC?
So at the moment, I'm working with a few groups which they are busy with doing stuff, but they're looking more on the natural product side of things. So because people are trying to go the route of precision medicine, where you develop drugs just for your country and for your people, that seems to be the route that most people are going now. Some are looking promising, but they still have to do it experimentally. So we have to see.
Excellent. No, thank you very much. So, but maybe we'll have 70 new people starting to look for such drugs from next week. But Krishna, there is a question by Victor, who is obviously outside South Africa. And he says, what software do you recommend for people that won't have access to Schrödinger?
Okay, you can do exactly the same stuff with Autodoc, which is open source, and in GROMACS is your molecular dynamics engine, which is also open source.
Okay. So GROMACS and what was the other one?
Autodoc.
Autodoc. Okay, perfect, excellent. That's fantastic. Thank you very much.
And yes, by the way, these are all available on the cluster, guys. So you all will have access to CHPC for those resources. So the MD can also be run on the GPU on CHPC.
Thank you. And I see that Patrick raised his hand. So Patrick, please, you're welcome to ask your question.
Okay, thank you, Prof. It's not really a question, but a commendation to Krishna and the team for organizing this series of programs. I think it's in the right direction, since it will enable more researchers to be able to use the tools that CHPC has. And I look forward to seeing this program continue within the weeks ahead. Thank you, Krishna and the team.
Thank you, Patrick. Thank you very much for that. Yeah. And thank you for, I'm very happy that, that there is an interest for this kind of courses. Excellent. There is a question by Adeshina, and it is, can docking be incorporated into higher dimensional quasars?
You probably can. But it's not going to be part of the things that we do. I also see we missed a question by Franklin. Will there perhaps be enzyme substrate interaction example? Franklin, unfortunately, I don't think we'll be doing any enzyme substrate stuff. We're probably just going to do protein-ligand, since it is a beginner course.
Yeah, but Krishna, maybe in in a second stage, we can do a more advanced course because it seems to that there is lots of interest. Yeah? So this month is just the appetizer and the introduction to the basics, and maybe a few months from now we can, if the interest is there, we can speak kindly to Krishna and ask him maybe to give us a more advanced course. Yeah, no. So thank you very much. Yeah.
And maybe I can look to doing the open source stuff. So I'll also be looking more into that to try and
that would be very useful. I think that would be really very useful, because then everybody can do it independently of the CHPC, and then if the need arises, then run the code on the CHPC machine. That's excellent. Yeah. I see Samuel ask if there is a recording. Yes, we are recording the talk, and we will post it later, so that maybe by tomorrow it should be in the NITheCS YouTube channel. Yeah, and we will share the link in social media, and then you will find it on the NITheCS website. Yeah. Krishna, I think there are a lot more questions in the chat. Do you still have time to go through them?
I'm fine with that, no problem. Let me just quickly see. Okay, so
there is one by, by Barukayat. Yeah.
So you made mention of protein coming with ligand. How do our ligand then bind to the protein? Okay, so you see what happens there is, if the protein already has a ligand bound to it, you're going to have to take the ligand out. The ligand being bound is just telling you where the active site is for that protein. So when it comes to docking, you'll remove the ligand and then you dock other ligands in the same place to see what happens. Okay, if I'm can we run NIME Schrödinger workflows on CHPC? Look, I think it's possible. I haven't tried it myself because I usually run the NIME stuff on my machine, but I think it is possible. I must actually have a look.
Excellent, thank you. And
Chemistry,
then,
guidance.
Yeah, there is a comment by Franklin. There are chemists here that need guidance because we do this type of things in the lab. Yeah, but I guess that's the whole purpose of this exercise, to give some guidance. Yeah.
So I'm hoping that this will at least give some initiative for this to progress. So we will look into getting into that maybe at a later stage.
Yes. No, as we said, this is just the appetizer. Yeah, just the introductory course, and we will look into more advanced stuff later. Yeah, excellent. And then lots of compliments. Yeah. Yes, we will share the link to the recordings. It usually comes automatically with the thank you that you participated, I think. Yeah. Okay, this is a Kanaim. I'm not sure how to pronounce it. Yeah. This is an interesting question by Sheikh. What is the success rate of CAD in drug discovery?
It's actually gone quite high now. If you consider the fact that if it wasn't for CAD, we wouldn't have a vaccine today for COVID. We'd all be still sitting inside, which we are. We'll be in lockdown still. If that was the case, we wouldn't have a vaccine.
Yeah. Okay.
Currently, what, 3 already. So.
You know, that's very nice. Then thank you very much for mentioning Krishna, because that shows exactly how relevant this topic is, yeah, and how relevant these methods are. Yeah. No, excellent. Thank you, Krishna. There are lots of compliments and thank yous. I will download the chat and share it with you so that you can read in peace all the compliments and accolades that you receive for your presentation. Yeah. So Krishna, thank you so much. Krishna also offered to share the slides. We will post them on the NITheCS website next to the announcement of the school and the recordings. And you should be able to watch it again tomorrow on YouTube. And please ask your friends and colleagues if they're interested to watch the video tomorrow, so that next week they can join us as well for lecture number 2. Yeah. So Krishna, thank you so much. It was really exciting. I enjoyed it very, very much, because this is a topic that I never really dealt with. So I'm really very happy that I learned a lot today, and I'm looking forward to playing with Schrödinger next week. So Krishna, thank you very much. Thank you very much to all the participants. And we hope to see you all again next week. And please inform your colleagues and friends to join us next week as well. Yeah. So thank you very much, Krishna. Have a good afternoon. Yeah. Thank you. A good afternoon to everyone.